What do you have next Tuesday? Test. So, a couple things I want to show first. This is a process that actually relates to both infection and relates to innate immunity. You already know what this is. It's called phagocytosis. I'll just give you a complete breakdown of it. Phagocytosis. You have a phagocytic immune cell. In this case, an innate immune system is your neutrophil, and it's often your monocyte that becomes, when activated and matured, is a macrophage or macrophage, depending on how you pronounce it. To a lesser extent, the eosinophils, not the basophils, because they only give off chemical signals to start inflammation. Phagocytosis, one of the first things you have is you can have a battle going on inside in your body. Sometimes it can be your cells are damaged and there are chemical signals sent out. Sometimes there are other immune cells fighting. Sometimes there are pieces, this is important, pieces of the pathogen because it's been destroyed. Won't be pieces of a bacterium, for instance, pieces of peptidoglycan floating around, pieces of capsule, pieces of fimbriae. All of those can be called something called PAMPs, which I'm going to go over to with one of the handouts in just a minute. Viruses, if they're broken down, can have pieces of capsid, maybe pieces of glycoprotein. So pieces of the pathogen, pieces of your own damaged cells, and also chemical signals can all be released from the site of the battle, the site of the infection. That causes something called positive chemotaxis. What does that mean to you? Taxis. That means the immune cells go toward the signals. What's negative chemotaxis? You go away from. So first you have positive chemotaxis. Then you're going to have everything else that's over here. You're going to have the cells coming in. They're going to see the bacterium. He's going to do what? He's going to surround it and bring the bacterium in what's called a phagosome. I may have different terms for this. I can say there's maturization and there's killing. Well, killing's obvious. Okay? What comes off of the Golgi body are the what? Lysosomes. Do you remember what's in them? Toxic oxygen byproducts, right? That could be hydrogen peroxide, sealant, oxygen, and others. You remember from metabolism how that tears up things? You can also have an acidic interior, and you have all sorts of enzymes for breaking down. All sorts. That's a lot, all in that one little vessel. Well, the lysosome is floating around, and the phagosome with the bacterium or the virus is floating around. They're both phospholipid bilayers. When they come together, they fuse. And you end up with what being dumped on the bacterium? All these products. What do you think happens to them? He's torn apart or dissolved or disintegrated, correct? This has a new name. They actually took the name of each separate vesicle and they squished the names together. And guess what they call it?
Phagolysosome.
Yeah, phagolysosome. There's one thing that doesn't happen in this process. Okay? There is no activation. Activation belongs to what type of immunity? The adaptive immunity. This is innate immunity. This is your macrophage. This is your neutrophil. He goes after anything, what do we say, that's non-self-patterned. He is not activated. He just attacks anything that's non-self-patterned. The B and T lymphocytes, which will come in the next unit, have to literally have presented and shown to them by another cell a pattern from the breakdown of a pathogen. So no activation. He's always on. He's always on. Does that make sense? That's one thing I want to go over just a little bit. Also, something that probably affects you. Remember our picture, our movie with the sneeze going everywhere? Okay. Let's say you weren't in the room and somebody somebody sneezed, or somebody had a cold and put their hands down. The process for you catching cold often goes like this. You put your hand down, you grab the handle. How many things do you touch during the day? How about the gas pump? How about your steering wheel, which you touch over and over and over and over and over, it's probably coated? How about the doors to go in somewhere? How about the handle to the water fountain? Have what on your hands? Probably everything, okay? But we're going to say this time, this time, cold virus. The other thing people do during the day, unconsciously, they rub their eyes, and they touch their nose. As soon as you rub your eyes, you're rubbing what? Viral particles into your eyes. Now, they don't have flagella, they don't have cilia, they don't have pseudopods, they can't hunt down anything, but you blink. And blink means you're, of course, keep your eyes moist so you have the right shape so you can see well, and also has lysozyme to help protect you from bacteria. But you're literally rinsing the viruses into another part of your body. Where are you rinsing them into? Your nasal passageways, your sinuses, and the back of your throat. Have you ever had that cough that always comes because there's a drip coming down the back of your throat? It's coming from where? Off of your nasal passageway. But what are you washing down this time? Not allergens, the virus. And if we're back to this, if the receptors match and he's bumping along your cells, eventually one of his receptors is going to meet one of the receptors he likes on your host cell, and he's going to attach. What did you just catch? You can really decrease your colds by not touching your face. Okay? But it's unconscious, so I don't know what to say about that. Good luck on that. But of course, washing your hands. That's not meant sarcastically. It's not sarcastic, is it? It's sarcastic. Okay? I hope it goes well. Now, we're back to innate systems. I'm going to show you a PowerPoint in a minute, so you're going to get to see some pictures of the defense cells. I'm going to start with one that people don't tend to like very much. It's the one that says non-specific chemical defense. I'm going to try to make it up on the top and very clear. It says non-proteins on the top. Non-proteins on the top. Let me check if I need this handout. If you do, I'll have some. I'll give you some. Help you out with that. So you might need a handout. Let's start at the beginning. It's on the back of defensive cells. It's on the back of, everybody's okay, good. That's very good. Okay, what I'm going to do here is I'm going to show you some areas I'm so interested in. Here's your list. Here's your list. Toll-like receptors, did you find it? Non-proteins, interferons, and complement. Complement I'm going to do with the PowerPoint. The other ones I'm going to do right here with this handout. Now what are these? These are not cells. They're chemicals. Okay, and complement happens to be a series of proteins. Toll-like receptors. Let me help you have an image of this. Remember you have the cytoplasmic membrane. We said there are some integral proteins. These are proteins. This means it goes all the way from the outside of the cell to what? The inside of the cell. Now here's the term I gave you a little bit of pre-teaching on, but I'll give it to you completely. Down at the bottom here, it says PANTS. That's a fancy word for pieces of pathogen. That's all it is. PANTS. Down here at the bottom, it says PANTS. P-A-M-P-S. All it says is pathogen-associated molecular patterns. Pieces of the pathogen. That's all you need to know. Don't worry about anything else because we already went what it could be. Pieces of the flagella, pieces of the cell wall, pieces of the peptidoglycan, pieces of the capsid, pieces of the pathogen. But, coming back to the toll-like receptors, I said the innate immune system is always on and it responds to non-self patterns. So, a floating piece of peptidoglycan would be a non-self pattern. We don't have any, right? There's none in us because we're not bacteria. In this case, that pattern can match the pattern of the outside of the integral protein called the toll-like receptor. And do you have a smartphone? You touch the smartphone on the outside, doesn't it cause something to happen? So the touch sends a signal from the outside to the inside of the cell phone or the inside of your immune cell. See, I really try to think of ways to get this across to you that are very familiar, because it's unfamiliar information. Several things can happen once that signal is sent because the PAMP, piece of the pathogen, has touched and matched the outside of the toll-like receptor. That's the 3-1 right down here. One, it can make that immune cell commit apoptosis. What is apoptosis?
Dead.
Yeah. Why would you want a cell commit apoptosis if it was infected with viruses? Other one, cannot cause death of the immune cell, but cause the immune cell, what I said for chemotaxis, I just sent over here, to send out more chemical signals into the bloodstream to call more immune cells to come fight. So to send out positive chemotactic signals, in this case technically they're chemicals or proteins, and those released from the side of the, well, from the immune cell says help us, we're sending these out, come help us. Does that make sense? So they're chemical signals. Okay, it's like the immune cells of a canoe in the middle of the lake and everybody starts throwing out balls and everybody, all the other people will come help up the stream of the balls toward the canoe. Last one right here, stimulate the adaptive immune system, which means help with the left side B and T cells, but that overlinks and that will take care of it later. Does that make sense? For a long time I didn't have the cell phone analogy, and I think it makes better sense with the cell phone. With a smartphone. It doesn't work if you have the other type of phone, which I had forever. I came in here with I like my old trusty phone. It didn't cost anything, okay? But people kept thinking it was a recorder, and it wasn't a recorder. And it worked for me until I needed to be sure I could do certain things. Non-proteins, similar idea. Similar idea. But look on the second sentence. These receptors are only on the inside for the PAMP to link in. Now that means there has to be a certain type of application. Toll-like receptors, you get the PAMP from the outside, trigger something inside. These non-proteins, they are only triggered from the inside of the cell they're in. So that means they're not going to be against so much bacteria, they're going to be against pieces of what's inside? Viruses. Cells infected with viruses, what is he making? Viral parts. And any one of those chunks out of those viral parts can come and touch the inside, couldn't it? And cause the cell to do similar type of reactions, commit apoptosis in some cases. Now, there is, they say, and this is research, the word research, I mean it's not known yet. They're wondering if there's a link to inflammatory bowel disease, but they also have about 40, 50, 60 other things that could be linked to that too. They don't have a good answer yet. So everybody okay with toll-like receptors and nods? Right? They're integral, well one's an integral protein, one's not. The integral protein has to go all the way through your immune cells. I'm going to move to interferons now. Interferons. Interferons are released by virally infected cells, host cells. They don't help the virally infected cell at all. Nothing. Nothing happens. But they're being released into the environment, which means the next layer of cells outside will have the interferon diffusing over to them. Interferon will link and of course cause reactions to the surrounding healthy cells. What they do to the surrounding healthy cells is they temporarily shut down protein synthesis in the healthy cells. Why would that be very important if you have in the center one of those budding, persistent, virally infected host cells, and then the next layer outside you have healthy cells, but they've all shut down the protein synthesis. They can't reproduce. Viruses can't reproduce. Viruses can come out from the center, whether it's lytic, break open, or whether it's budded out and the original cell is still alive. They go to the next healthy cell, they always go to the next layer out. They can attach, they can enter, but the protein synthesis has been shut off. So they can't do what? They can't make viral parts. Can't make viral parts, they can't continue their infection. And for a while these cells stay turned off Eventually the virus can't keep control forever, and then later the cell hopefully will turn its synthesis back on and it'll be fine, and it outweighted the virus. It's not as beautiful as it sounds, because don't we all still get colds and everything else? Yes. They once thought when they first discovered interferons, they got the magic cure for all viruses. To a degree they can help, to a degree they don't help, depending on real life. Two types, type 1 and type 2, and after this I'm heading for the PowerPoint. Type 1 is our innate system. That's your poor infected macrophage, okay? And then, of course, you want to prevent healthy cells from being infected. Type 2, we're going to push over more to the adaptive system, and we'll go over that a little later. Is everybody okay with this? Because they're odd names, toll-like receptors, non-proteins. But it's really very simple what they do. You can get some very complex little we call films that come up on some of the internet, but that's the core of what they do. Okay? All right, we're going to now you have a break, and you get a humorous example. They put a new screen in the other room. It stays down. And I'm clicking and when it fails, that's not good. I thought I had it. I don't want to tie it because it'll make a knot. And the next person will be able to get it up. Now, see? Humor. That's humor. Every professor should have to do that because it humbles you a little bit. Okay? Everybody all right? Coming to the PowerPoint. And don't forget this, because I'm going to go over this at the beginning. This is your map. What we just did were what? Toll-like receptors, non and interferons, right there. We just did that section. Right here. Okay, coming to the next. As it wakes up. Innate immunity. I'm going to turn the lights off for some of this. I want you to see some of these cells. We already talked about the fact that just having a bacteria land on you doesn't mean you're infected. You already know that. Know what species resistance is, but you've really been taught this many times. A plant virus can't bother us because why? Different receptors, our body temperature is different, everything's different. Everything is different. So, organisms that attack plants don't attack people. Organisms that attack people don't tend to go out there and attack plants. Does that make sense? Okay. 3 lines of defense. We already talked about skin, which is the physical barrier. Innate immunity we just started, and adaptive immunity, excuse me, is the next section. You already know about the stomach. You already know about lysozyme. Some of this is a repeat. Some of it is a somewhat of a review. We already talked about this because that was skin. We already talked about dendritic cells. What were they? Underneath every boundary, your skin, your intestines, your respiratory mucous membranes, you have a cell underneath that has the ability to phagocytize, called the dendritic cell. It is the scout. It will come phagocytize the first organism that's hitting you from the outside, and it will do what? Break it down into patterns that it's running for your lymph nodes. It's going to help start the adaptive system. Adaptive system takes 2 or 3 weeks to start responding. Since it takes so long to respond, what you want your dendritic cell turning it on as fast as possible. In the meantime, what's keeping you safe besides your skin? The innate system, which is always what? Always on. Attacks everything that's non-cell. Okay? That's a dendritic cell. It will send out pseudopods. It will take in a piece of any invader. And he's hunting to find the patterns to go present to the D cells. Alright? These are just some pictures. This is more of a story. We already said that. There he is presenting. This is more the adaptive system. He's taken the pattern, he's broken down the bacterium, and he's presenting the pattern to what will later be adaptive system to turn on the adaptive system. You don't have to know this right now. You will see it again later. Okay? Remember the immune cells. I'm going to give you pictures of them. Erythrocytes don't fight anything. What are they? Red blood cells also transport. Platelets just seal you off. They're clotting. Right? Keep you from bleeding to death and keep more things from getting in. Here we go. Basophil, inflammation only. This is straight from the book. Neutrophil often attacks bacteria, usually the first one to the site of infection. Eosinophil can do some other kills, can be phagocytic, but often means worms, parasitic worms. Monocyte becomes the macrophage. He's got Zilla. He attacks everything. He attacks the bacteria. He attacks the, you get a splinter, the dirt. He attacks pollen if it's in you. He attacks the wood of the splinter. He attacks everything. He's what?
Slowest to get there.
I'm sorry, one more time.
The slowest to get to it.
Oh, slowest? It depends on your definition of slowest, because there's two types, so there's several types. There's the monocyte that's going through that has to find the chemicals, squeeze through the tissue, and differentiate. Then there are ones that are already they're not activated. The ones that already met a pathogen, they're called wandering macrophages, and they just sort of patrol your whole blood system. If they happen to be right there, you're all right. Now remember, I teach micro. Nursing takes a different slant. When nursing wants something, you give nursing what nursing wants. Don't confuse it. Okay? Lymphocytes, the adaptive system. B and T cells. And they look just alike. Just alike. We already talked about this. That's a neutrophil. I am not going to ask you, I'm not going to put a picture up and ask you to identify it. I'll leave that to nursing. Okay? I will want you to know what they can do and how to protect you and how to deal with bacteria. Eosinophil, okay? Granulocyte or agranulocyte? Granulocyte. Can attack worms, can also, we'll get this into another chapter, can help start an allergic reaction. There's a poor little innocent worm, okay? Now, eosinophils cannot swallow the worm. They come up to them and they drop what on the surface? Toxins. They drop toxins. They can't swallow him, he's too big. But they'll still kill him. They'll still kill him. Okay, basophils, we said not phagocytic. Inflammation, of course, inflammation means you get weaker blood vessels, etc. So basophil. Lymphocytes, I do want you to know natural killer lymphocytes, NK. They will go up, and if there is an antibody on a pathogen, they will kill it. But not if it doesn't have that antibody. Lymphocytes look all the same. Agranulocyte or granulocyte? Agranulocyte. Monocytes, think Godzilla. Everything. Okay? Monocytes become macrophages. This is not really the best picture, because they can often be a little bit more amorphous than this. Okay? Wandering ones are the ones I was talking about. They can just keep flowing around and around in your bloodstream. Fixed ones will sit and wait in your lymph nodes for something to come by. And that's back to when I started. Lab tests we already talked about. Phagocytosis we did. Yes?
What was it for the neutrophil again?
Neutrophil is phagocytic. Often, no, I'm not saying always, often the first one to site infection. Usually goes after bacteria. Alright? These poor little bacteria are going to be eaten. And they're going to be phagocytized. Remember, lysosome comes over, fuses with phagosome, either be phagolysosome, you have the products dumped on it. No activation, just anything that's non-cell. Activation belongs to adaptive. Okay, how do they recognize non-cell? Microbial parts are not part of you. You don't have fibrin inside you. You don't have the type of DNA that they have physically methylated at different places. You don't have peptidoglycan in you. You don't have the same type of protein as their flagella. Not found in the body. Non-cell. Of course, this does cause a problem that overlaps with allergens, which I'll get into later. That's still not cell. Okay? Opsonins is one thing that overlaps with the adaptive system. I'm going to go ahead and tell you this because we're heading for complement. Complement. Complement, if you look at that page with the non-specific chemical effects, it's the one, oh, my notes in the way, I'll get my notes out of the way. It's right down here. Right down here. Complement is a series of 20 proteins in your blood. They always act in sequence. That's sometimes they call it the complement cascade. This is from, this will be on the focus notes, so we're not going to hand out. We're on the focus notes, not on the handout. Which means 1 goes first, 2 goes second, 3 goes third, get the idea? 4 goes fourth. They have to happen or fall into place in order. Not that simple. Because it could be 3A has to link to 3B, which links to 3C, while 3D goes somewhere else over here and also attaches to the pathogen. I'm not going to make you know that. What you have next? Just know 20 proteins act always in the correct order, and some of them will link to each other. Okay? Now, an opsonin is a very fancy word. A metaphor for that, or an analogy is, think of it like a fish hook or a handle. We said, for instance, that Streptococcus pneumoniae has a capsule. Capsule makes it slippery, and it can slide away. What some immune system, and this includes some of the complement proteins, does is stick a handle on it. Put that complement protein on it, or put it on like a fish hook. Now the immune cells have something to grab. They have something to grab. Opsonin can be like a handle or a fish hook. It can also be like a flag or a signal. That's the two purposes for an opsonin. If it's a signal, what do we say about capsule? It can appear as a generic sugar, and we think it's the same as cell. But if a piece of complement sticks onto it, it's marking it as target. Remember all those things they used to say where somebody went in and they had the laser? You've seen the movies and somebody has a red dot on their forehead? They are a target. Somebody's going to shoot them, right? Unless they get out of it in the movie. Opsonins are the same way. So opsonins have two purposes. One, they can take a pathogen, the piece of complement, and also happens to be antibodies, but we're doing that later. Piece of complement can attach to the outside of the capsule or other pathogen, marks it, says destroy what I am on. Signal, flag, or maybe a laser point. What's the other one? Help them attach and grab. Does that make sense? There's two reasons. One, help the immune cell get a handle on it. If you take this thing and you put a fish hook on it, and the immune cell grabs the other end of the fish hook, maybe with a line and pulls it in, what has to come in on the end of the fish hook? A pathogen. Pathogen has to come in. So handle or fish hook, 1. 2, identify to attack. I can't think of any other way to say that. Complement is a series of 20 different proteins. And you probably will do your focus notes for this. And they can do that. Complement lands on the pathogen. They can say, attack this pathogen. Attack it. Alright? Now, I want to say one more thing about complement. Don't have it in here, where else? I'll come back to that. There's my complement. We're going to come back to this. It says sequential order, cascade effect. You can also do other things with complement. Not only as an opsonin, which we just discussed, it can be a positive chemotactic factor, which means if they're in the area of infection, we're telling what to come. What do you want to come to the area of infection? It's immune cells, all your immune cells. They again can trigger inflammation, just like the basophils can. You get tired of hearing that. Basophils can trigger inflammation. Complement can trigger inflammation. The cell that releases chemotactic factors can trigger inflammation. So in other words, a lot of things trigger inflammation. Okay? This is different. Direct killing. It's called MAC complex. Okay, that stands for, we have to have a fancy word for it, membrane attack complex. This is where all the jigsaw pieces have to finish to the last one. Complement's about 20 proteins. Have you ever done one of those 3D puzzles? Not a flat one, but a 3D one? You gotta put all the jigsaw pieces in? I can't say I have. I never had a good time to do it. Think of complement like that. Except it's forming a donut puzzle. It's forming a donut on the surface of the pathogen. First complement jigsaw piece goes in. Second one goes in, links to the first. Third goes in, links to the next. Every last piece of complement has to fall into place. Period. Every last piece of complement has to fall into place. And you have to have the complete jigsaw puzzle put together for the donut on the surface of the pathogen. Once the last complement protein drops into place, the donut opens up a hole into the pathogen, makes a hole. And we're not talking one, we're talking hundreds, maybe hundreds and hundreds of holes being made into this pathogen. What's going to happen to the insides? The whole thing is going to spew out and the pathogen is going to die. So that's called the MAC complex because it's making a hole straight through the what? The cell membrane and the cell wall. And if all the insides are leaking out, what happens to the pathogen? Dies. Won't work if all the complement proteins don't drop in sequence and if the last one doesn't come in. If the last one doesn't come in, the whole won't open. Okay, so complement has a lot of things. It can be an opsonin. It can signal attack this. It can start inflammation. It can also give these sort of positive chemotaxis chemical to say more immune cells come. And if the entire complement system falls into place, making a donut, it can open up holes into the target and actually complement can kill it itself. Does that make sense? It's a really neat system. And that's just from the full cascade. And it says gram positives tend to be resistant. Why? You know this. You know this way back when you used gram positive and gram negative cell wall. Gram positives have a very thick layer of peptidoglycan. Maybe you can put a hole down to here, but it's hard to do what? Get a hole all the way through. Does that make sense? Plus, they don't even have any cell membrane on the outside. They say complement all acts in the same way at the end. Opsonin, positive chemotactic factor, start inflammation, MAC complex, if all the pieces fall into place. Okay? But complement starts in 3 different ways. It's like if all of us go and we pick different ways to get to McDonald's or Burger King, we all start from a different way, we still all end up at the same spot. So it all does the same thing, but the 3 different beginnings are called this. Classical means an antibody had to start that first complement protein starting the sequence. Classical antibody that comes from your adaptive system starts the cascade, starts the dominoes. Think of them like dominoes. Something has to start the first domino. For alternate, it's going to be what? Piece of the pathogen. Piece of the pathogen. That's sort of easy. We already talked about those causing immune cells to respond. Last one, it simply sort of stays toward just the polysaccharide, which means polycapsule, and it's called the lectin. But it's still, if you have those, remember all those pictures of dominoes and you hit the first one, the whole room starts to go? It doesn't matter what pushes the first domino. They're all going to go down. Okay? Does that make sense? Okay, and then we're going to go back to our cells into a different way. Take a deep breath. Who are our cells? Basophils for inflammation. What you going to say? Okay? Maybe some allergic responses are given to those cells. Now we also have non-phagocytic killing, but in this case, we're not talking the macrophage. He is only phagocytic. We're talking the neutrophil and the eosinophil. We've now focused down to the neutrophil and eosinophil. Are they phagocytic? Yes. Okay? They can also do something else. If you're heading for the eosinophils, I said they are not as phagocytic as neutrophils and macrophages. They like to kill by a type of membrane, well, type of microbial trap, a trap. What they do, well, this is the worms. You already got the worms, so I told you about the worms. We're not going to go over the worms again. Talk about extracellular toxins killing. Here's where I want to go. It can also kill bacteria, especially gram negatives. Two places in a eukaryotic cell have DNA. The nucleus with the chromosome, and the powerhouses, which are the what? The mitochondria also have DNA. So they will take, remember how DNA is packed tightly. This is an exaggeration. If you took a ping pong ball of DNA and released all the stabilizing binding proteins, the DNA would fill this building. It's like a huge net, though, if you look at it from another point of view. In this case, the eosinophils will release the restraints on the DNA from their mitochondria. Release it out into the environment. They're really releasing a what? A huge net. A huge net. Of course, they're at the site of infection, so they're probably going to catch more, right? So they're trying to catch fish. They may put some proteins with it. It is a physical barrier. The bacteria get tangled up. They think some of the proteins may kill the bacteria just being held there long enough to starve to death to kill them. And they're not quite sure how it kills. So they throw out a huge net. But don't call this net, because net is actually only the word you use for neutrophils. It's another word for net. Here's another word for net. Lattice work. A huge lattice work. Don't use the word net, because that actually means neutrophil entrapment thing. But that's what it is. Everybody okay with that? Neutrophils. Yes, very phagocytic. Very, very, very phagocytic. They can kill with toxic oxygen byproducts and simply release them on top of the invader, okay, and kill. They can use, this one is called NET. It stands for, well, what they do, neutrophil entrapment section. They don't use the DNA from the mitochondria, which is somewhat limited. They use the DNA from their nucleus. How much more DNA is that? It is huge on a microbiology scale. So they release all the DNA from their nucleus out into the environment, trap everything. What is it going to trap? Everything? It's going to trap whatever's in there, unless it's big enough to get through, like a worm. And it's going to kill. They do have some types of materials in there that can help kill. They have toxic oxygen byproducts. The net comes out. And they have some antimicrobial peptides. Bacteria die. Yes?
You said that stood for neutrophil enactment.
Entrapment.
Entrapment.
Just don't even bother with that. Just know net. My concern is the two terms for net. When it's N-E-T, capital, all the way across, it has to be the neutrophil. If it's N, little e, little t, it just means a net, like a fish net. And the eosinophil puts out a little one. Okay? But if you see it capitalized all the way, that has to be the neutrophil, which has the bigger lattice work / net, because it releases chromosomal DNA versus just the mitochondrial DNA. Does that make sense? And let's give you some pictures. That's a net. Eosinophil is as in capital N-E-T. It's releasing his chromosomal DNA. He's going to trap gram positive and gram negatives in this, and they're going to die. What do you think happens to a neutrophil? He dies too. So, some neutrophils, and most of them are phagocytosis. They live, they keep breaking down things. Some put some toxic oxygen byproducts on the surface of the target, they kill. Few decide they're going to go commit suicide, okay? But they're taking as many of the enemy with them. They get in the center, and they take as many of the pathogen as they can with them. Does that make sense? And they don't come back. They're gone. That is an electron micrograph of the DNA being released as well. Okay? That's the poor little innocent bacteria being caught up in the net. I don't actually live with bacteria, but that's the idea. See it? NK lymphocytes. It says I ask you to know one lymphocyte. NK stands for natural killer. We do have them with the innate immune system, but really most of the lymphocytes are in the adaptive system. And yes, there is an overlap here. You can't quite tease these apart. They go after virally infected cells, which also I told you to know the TC cells do. Okay. They secrete little toxins on the surface. Does that sound like eosinophil and a worm? A little bit. They have markers. There are markers on that virally infected cell or tumor that tell them to attack it. I'll tell you that marker right now is the antibody in most cases. Maybe others. So that has to come from the adaptive system. So they have to have a marker. What's that marker called? Opsonin. The antibody is the specific one, yes. They have to have opsonin to say attack this. And they of course go after virally infected, and yes, there is some sort of overlapping range of cancer cells. Does that make sense? Mm-hmm. Now when we get to the TC cells, I have a whole system of them to teach you.
TC cells or T cells?
TC. That's not the same thing as things. Capital T with a little c at the bottom. And I'll tell you what that means in adaptive system. C is in, I don't want to say cancer, that's bad. Kit Kat. No, that's K. I'm drawing a blank. I don't know. Cat begins with a C. Okay? I don't have to spell cat. Right? So NK lymphocytes are sort of being plopped here. They need an opsonin. You know what the opsonins are. Complement can be opsonin. Antibodies are the other opsonin. And tells them to attack this. In this case, the target is virally infected. Everybody okay with that? All right. We did these. We did the complement. It's too much information here. People start learning things that I'm not trying to focus on when I put too much information. So try to focus. Inflammation. What have you heard? Complement starts the inflammation. PAMPs can, what? Basophils start inflammation, right? Okay. You can also have inflammation started from the toll-like receptors being activated on the immune cell that sends out chemical signals that help start inflammation. What starts inflammation? Many things. What is it? More permeable blood vessels. Tells the phagocytes to come to the site of infection. We'd rather have 1,000 fighting for you than 10. And of course, it can also help with some tissue repair. You get swelling, redness, okay, edema, which is the swelling. You get some sort of itching and pain. All that means is that the ends of the nerve endings are made to fire. You do know that itch is the same thing as pain. There is no difference between an itch and pain, except for how fast that nerve cell fires. The itches is firing less. Its pain is firing more. And they're both warnings. But don't scratch yourself in the itch because you're going to your skin. Like that. What? You get infections.
I understand the kissing bug.
Well, you still remember the kissing bug. That's going to stay with y'all, isn't it? See? Even unconscious itching. That'll get you. We already talked about this dilation. I'm not going to do a handout because it's word there. Causes the monocytes and neutrophils to come to the infection site. Monocyte is macrophage. Keep them linked together. Okay? Now I said neutrophils usually arrive first. We already talked about diapetesis. That's going to be what macrophage or neutrophil squeezes through, usually within an hour. And then of course he's going to eat absolutely anything. What is pus? Broken down dead cells, new cells, pure cells, broken down pathogens, everything in the battlefield that's sort of been left there, the bodies have been left there. Okay, got pieces of. Okay, dead neutrophils, etc. I'm going to come back to this in a minute. What is not involved in inflammation? It's actually a system and or process to help protect you. It's an immune system to help protect you. There is not necessarily any sting. because that's not going to happen unless you start rotting or there's a really serious secondary problem from it. Plain old inflammation is that bite, that's red, that itches, that you'll scratch, that brings the immune cells and can itch to hurt. Does that make sense? If we move to the next stage, you've got something else going on. Right? Here's the point. Chemotactic factors, positive chemotaxis. That means bring them to the site. Bring them to the site. We said complement proteins, bring them to the site. Leukotrienes is a fancy word for some of the chemical signals that are released by the immune cells. Previously we said, with the toll-like receptors, immune cells can be triggered to release chemicals to call what? Other immune cells to the side of the fight. One of the names for those is leukotrienes. That's one type of chemical. I said microbial parts, we called them, the book calls them PAMPs. Toxins are still non-self and can still cause positive chemotaxis. Okay. Are you ready to head for fever? Fever is not completely understood. It can help you. It can also, in some cases, harm you. You ever had to take a child and put him in lukewarm water in the bathtub and get his fever down? Yes. You don't use cold water. Don't put the kid in ice cold water with ice cubes. Don't hurt people do that. No. You don't want to shock him. Fever, there's the primitive part of your brain, the hypothalamus. Guess what reaches the hypothalamus? It overlaps with pancreas. You'll see it right now. They call these PAMPs, though, pyrogens because pyro means fire and it makes you hot. Okay? They reach, and here's what I'm going to talk about right here. Does this sound familiar? Toxins, bacterial parts, those are just the same as PAMPs. Alright? A little bit different, also can trigger fever, i.e. is a pyrogen, antibody-antigen complex. Let's use the definitions. An antigen is any pattern from anything that causes your immune system to attack. An antigen is any pattern at all that causes your immune system to respond and to attack. It can be a piece of bacterium you're going to defend yourself. It could be peanut butter, which you are not have an infection, you have an allergy. But it's seen as non-self. Antibody is that little Y, which we're really going to get into in the next chapter. The top of the two parts of the Y will link to the foreign pattern, which in this case is the antigen. That's where they always link. Top of the Y from the antibody links to the foreign pattern, which in this case we're saying is bacteria. Could be pollen. Okay, and of course, any molecules released by the phagocytes that ingest in the microbe, what's that our first fancy word for saying? Some of the leukotrienes are chemical signals that are given off from the immune cells that are fighting, telling your fever to go. Someone, we don't have a fever. And you know what fever is. You shiver, the blood vessels on the outside constrict because your hands get colder, but not on the inside. So it's really outside versus in is a little bit different. Okay. Supposedly, they're not quite sure what fever does. They think it helps set the body temperature so the pathogen can infect as well. They think it helps stop viral attachment or cycle somehow. They also think it helps some of the interferons work better. That's not saying they think. So that is still in the process of being checked. And to some stage, stage crisis of fever, they don't know what it does. Okay, then of course you cool off, as you already know. Say this, inhibit growth of some microbes, enhance immune systems such as interferons are here, and help in ways they don't know. If the fever stays too high for too long, what happens? Brain damage. Can't let it stay too high for too long. Many doctors, though, tell you to let a what run? A low-grade fever, let the low-grade fever run. Okay, this has been going on for like 3 years. It's been balanced everything. So, for a while, I've learned to qualify almost everything I say. Somebody says, I've had a fever for 8 months, am I okay? First, go ask the medical doctor, don't ask me. All right? This is what we said, interference, and that was that one. Now I'm going to simply do a review. Any questions on that? Okay. I'm going to briefly review this page with the GPS map of the native immune system. Then I'm going to go back to the beginning of our, does anybody need one of these? Yes, I see it. Raise your hands. Okay. You want 2 more?
I don't know where
I'll pass this one down and over. Anybody else? Anybody else? This is what we just did. Put this back. And as I said, I tried to make a system so you can look and see it all broken down in one place. Innate immunity, right over here on this side, resident microbes, how your skin protects you, and also something about your mucous membranes. Over here, cells and chemicals, here are your cells, right here. We already did that. Our natural killer lymphocytes, we say kills with toxins. We said it has to have an opsonin, it goes after virally infected cells. We talked about the macrophage. Okay? Over here, we mentioned inflammation, of course that's the basophil. Phagocytosis, I just wrote it on the board. Think about it. Phagocytic means it's able to eat the cell. Chemotaxis means positive chemotaxis. That means those chemicals, just such as leukotrienes, are released and immune cells come. You have to adhere, they have to be able to grip the target. They then ingest it, and I'm just going to throw them all together. Ingest means they put it into the lysosome, break down and kill, and eliminate means they spew out the pieces of the pathogen as waste. They just spew it out as waste. There is no what here? There is no activation, because it attacks anything that is non-self-pathogen. Complement we went over is an opsonin, it's positive chemotaxis, can start inflammation. There's your MAC attack, and it mentions a couple of ways beginning of the pathways that we talked about. Coming over here to systems, we already talked about inflammation, okay? Permeability, migration of phagocytes. We talked about iron wars, which is way over here by itself. What are the 2 organisms that want iron? Staphylococcus. Who's the other fellow? Neisseria. And fever with pyrogens. Pyrogens pretty much overlap with PAMPs with the exception of the antibody-antigen linkages or complexes. Okay, does that make sense? That's what you just did. That's the overlap for everything. Okay, first, before I start the review, is there anything y'all would like for me to review more than another subject? Anything you'd like me to review more than another subject? And look at your notes. Tell me what you like. I'm going to try to untie this. Okay, and then I'm going to start with viruses. And I will respond to you later if you have a question that you come up with while I'm going over this. I'll be glad to change course. Viruses. And I will tend to cross-link information when I do reviews. They are acellular. What does that mean? They're not cells. Can they hunt you down? No. They have to bump into you. They bump into you. Receptors have to match you. Receptors have to match you. If we're talking a bacteriophage, that's a virus that only affects what?
Bacteria.
Bacteria. Now, he has a different core or relative structure than animal viruses. There's something that can be in animal viruses that cannot be in a bacteriophage. What is it? No, bacterial can. That's only on bacteria. But it's okay to try. Okay, let's give you some more memory jogs. Okay, let me do some memory jogs. I'm not trying to put you on the spot. I'm trying to help you. Core virus. DNA or RNA inside, correct? Then you have a shell made of what type of structure? A capsid made of proteins. That's it. That's as far as it goes for bacteriophages. Now they can have a complex shape, which means like the polyhedron on top of the tail, on top of the tail fibers, but that's the core shape. If you step over into animal viruses, some do have and some don't have an extra layer outside the capsid. What can it? Envelope. What does the envelope actually consist of? It's a phospholipid bilayer. Is it active, relatively speaking? I mean, where did they get it? They stole it from the host cell. They can steal it from 3 places when they bud out. What 3 places? Regular old cell membrane. What's the other? The nuclear membrane. And the endoplasmic and reticulum, DUR. Okay, going back to bacteriophages. And this is how I taught the concepts of lytic and lysogenic. First, all viruses go through 5 steps. Attach, enter in some form, and what? Synthesis and replication, assembly, and what's the last? Release. And it can shift a little bit on enter. For bacteriophages, there's only one way to enter, called direct entry. But what does that mean? The DNA is sent in and what's kept outside? The capsid is left outside. For animal viruses, there are 3 ways to enter. One is direct entry, still. Leave the capsid outside, what into? Membrane fusion, and where? And endocytosis, and I'll also accept phagocytosis. There's actually turnaround immune cells against us, isn't it? So we just talk about phagocytic immune cells. Viruses sometimes won't be. It's not good for us. It works for them. Okay, back to after we do the basics there. Lytic means the entire cycle occurs. In that case, we're talking about the release here. How is the release with a lytic cycle T4phage? What happens to the host? Actual cracks open, breaks lysis. Lysis means break open. Breaks open the host. Now, if you move to a lysogenic phage, such as lambda, yes, it can go in. Yes, it can take over. Yes, it can force parts to be made. Yes, it can force assembly, which will be lytic, or it could do what? It could go into the host chromosome and quietly stay inside the host. It's not lysing that cell, is it? But it does pass the virus genome on to something. All the offspring. This may not be just children, grandchildren, great-grandchildren. This may be for the next 40,000 years. Get the idea? I mean, very paired out. Of course, there's nothing against some other cancers and some other tissues. If that lysogenic virus is triggered, what's the word for trigger? Oh, that's the wrong word. Induction, or to induce. Induction or to induce. What are some methods to induce or to cause induction of a lysogenic virus to become lytic? The what? Some genetic might, but that's often goes go to chemistry, such as chemicals, also radiation, UV, X-rays, all of radiation. Okay, chemicals lead, preservatives, aflatoxin. Well, you can have the peanut butter today. All right, now the organism has been switched from lysogenic to lytic. Is now going to do what to the host cell? It's going to crack it open. give me two examples. Well, first let's back up a step. Lysogenic means it has to have a host that is a bacterial. What is the term if the target host is animal cell? Latent or latency. And in that case, there could be slight differences. The genome of an animal virus may stay independent of the chromosome or it may incorporate. Now let's move over to animal cells with latency. Give me two illnesses where you can have a lytic and flip between lytic and lysogenic cycles.
Diphtheria.
Not diphtheria. Diphtheria is a toxin. That's a lysogenic sponge in the center. Yeah, great. Not penicillin, it's antibiotic. Okay, chickenpox. What happens with chickenpox? Probably didn't ask the question all that. Chickenpox goes in, you get chickenpox, you have a vaccine. Becomes what inside your cells? Latent. Immune system starts getting weaker or some sort of induction trigger. Because I've heard of children that have children that shingles too. And you get shingles. What's the other one that some people have these little places show up on herpes? In which case it's fever blisters. So, latent, quiet, you're calm, you're at the family reunion, you're having fun, your friends are all there. Suddenly you have a terrible microtest. Your blood chemistry does what? Shifts. That can be induction and cause the latent herpes to become fever blisters. That's lytic. Lytic because what did it do to your skin cells? Cracked them open. But you're not cured of it because two things. One, you still have some that are still left back that are still latent that didn't trigger. Not all of them triggered. And also, all your dissolved skin cells, all those blisters, released what? More virus. And it went to the next skin cells that were close by. Does that make sense? Okay. All right. So, what are the three shapes for virus? Polyhedron, helical, and the one that catches everything else, complex. If you have, trick question, if you have a virus that has gone into your host cell, there's only there's a core way it exists. A virus inside your cell, the core before it does anything is what? It's just the DNA or RNA. An animal virus that goes into a person, if it goes in with membrane fusion or endocytosis, has a capsid, but it can't do anything until it's broken. That capsid has to be what? Broken or removed, called uncoating. So the actual piece that controls capsid's destroyed, envelope's left behind. What is really the virus existing as inside your cell? In that case, it would be RNA, nucleic acid. That's all it is. Taking over our cells. Taking over our cells. Okay, if I say budding, what do you think of? Budding. Well, not. That's where Michael gets me. There's the same term in two different things. I worked so hard to try to talk it out. Yes, yeast. But let's put it into virus leaving infected host cell.
Just go to the bilateral.
The virus goes through like flu, rips off the membrane, goes out, but the host cell's left alive. I like the yeast cells, but okay. Now, let's see, let's do the next one. Can you tell me the parts of the influenza virus? Start from the inside. RNA. Then what? Shell is icosahedron. How many phases? 20 capsid made of protein. Little beads that would make up each of the triangular faces. That's where the concerns, that's called the capsomere. Okay. Then another layer, like having the core of the earth and then the next shell outside, is the envelope. On the envelope is the glycoproteins. What are the two letters used for influenza glycoproteins? H and N. Glycoproteins are attachment proteins, right? They help the organism attach, attach. How does he get the glycoproteins? Does he make them inside our host cell? And pulls them along with them outside as he leaves? No, what does he do? He makes our cell membrane make the spikes on the cell membrane. Then when he goes and buzzes through the cell membrane and rips off the envelope, what has he made us form for him? The glycoprotein spikes. And they're in the right place. That's pretty smart, isn't it? They're already in the right place. Not good for us, but that's okay.
Does that make them recognize how they want to stop?
That gets into some interesting things because that can be a non-self pattern to tell the adaptive immunity system something's wrong. And they may know to come working with it to heal. Give you the idea? That's very smart. Because that's a non-self pattern on what? Our cells. With the exception, if he starts drifting, antigenic drift, you can actually change some patterns that get closer to pigs, that get closer to, pigs have close antigens to who? Humans. I'm not trying to knock pigs. Have y'all ever heard or seen a full-size pig? We're not talking pig. We're talking back here, okay? Pig goes around here, you can't control it. Have you ever heard one of those when they make noises in a farm? It's Halloween's coming tomorrow, right? The sound of all the things out for devils and demons to me sounds just like a huge pig. I one time went, I was very young, went past a pig shed. I didn't know what was in there, and it sounded to me as a little girl like, oh my gosh. Then of course we looked, and those things will kill you. Those things can tromping you down underneath them, they can kill you. Okay? If they have a bad attitude. See, people think I call it grocery store thinking. Everything's in a perfect little modest size. It's all clean and it's all on the shelf. That's not the real world. Certainly when it comes to pigs, you can smell them all the way too. Okay, now, because of the excrement, we're going to try to get some things done on inspection. See, I woke you up. I hope wake you up, wake you up. I really wanted to show you a movie, but I'm not going to have time to do it. I have a great movie of a neutrophil hunting down, and maybe I get time in my app to do it, hunting down and killing some just innocent little staph. I want to show you that movie. Do y'all want to review on the movie? Review. Okay. Don't forget okay, we're going back to infection now. Virulence factors. Virulence factors are factors that help make you sick, help organisms establish infection. Toxins. 2 types of toxins. What are your 2 types?
Endo and exo.
Endo and exo. One is the structural part of only the gram-negative outer cell membrane. Endo. E-N-D-O. I'll give you a tripping stone. People, when it comes, you know, sometimes there's a little bit of matching on my test. Sometimes people confuse enterotoxin with endotoxin. ENDO is the lipid A, which is in the gram-negative cell wall, or cell membrane of that. Entero is one of the 3 types of exotoxins. Exotoxins are not structurally part of the bacterium. They are small, often proteinaceous forms that are excreted from the bacterium into its environment, which often is toxic. 3 types of exotoxins. Neurotoxins. Cytotoxic, which breaks the cells down, and the entero, which heads for the intestinal area. Everybody okay? I can say it again. Is it okay?
Neurotoxin is the first one.
Okay, I'll say it again. It doesn't have to be in certain order. Cytotoxin, break down the cells. Right? Neurotoxin, hit any of the neurons, all including going after the brain. The last one is intestines. Think of E-N-T-E-R-O, enterotoxin. Okay? I promise you, when y'all ask me a question, you're helping people. That's one set of virulence factors, the toxins. Of course, the bacterium can sit here, release the toxins to your body. You have toxemia and you're sick throughout. What do we defend against toxins with? Often antibodies. Let's move to enzymes. Enzymes as virulence factors. I gave you a couple of examples. Hyaluronidase. What does it break down? Hyaluronic acid, which is sort of like the biological mortar, like the mortar between bricks, but the bricks are your cells. Biological glue between your cells. If the bacteria can break that down, he simply infects further. He gets through that. Collagen is what? A major protein that helps build part of your tissues. If the bacterium produces collagenase, it's just flat out breaking down some of your tissue. The next I sort of teach right now is a set. Coagulase, don't mix it up with collagenase. Coagulase causes coagulation. Who hides inside that coagulated shell? Real bad for doing it. Use bigger paper. Staph. Staphylococcus. He's hiding inside because he wants to increase his cell numbers and he's hidden from your immune system. He then produces the other enzyme that helps, kinase, that helps put holes in the coagulated shell so you can get out and infect you better. So that's toxins, that's some enzymes. I left one enzyme out that ringworm likes to use. Fungi, right? What's the name of it? Keratinase. Keratinase, that's good. That's what's our fingernails made of. Right here. Here. You just flat out break it down. Break it down. Now, you can also have anti-phagocytic factors, which help them invade. One of the big ones is capsule. Capsule has three things it can do, which I sort of already taught at the beginning. One, can help hide, because it's a sugar, and it might be seen as self. As you just learned, you can put like sauna on it so it can be grabbed. Can also be slippery like soap, slide away. And the one that overlaps with the virulence factor called biofilm sticks. And then everything else sticks, and you keep getting worse and worse and worse and worse. Okay. Now, we're going to the chemicals that can be anti-phagocytic. First one, Neisseria gonorrhea secretes chemicals, and this relates to phagocytosis, that don't allow two very special doesn't allow the phagosome to fuse with the lysosome. If the lysosome never fuses with the phagosome, then Neisseria is in the phagosome. Nothing happens to him. He's fine. He's sitting in his little house with a balloon and he's okay. Strep throat has what? M protein. That helps resist phagocytosis. You can also have Fimbriae and some bacteria that help resist phagocytosis that overlaps with Neisseria. Last one doesn't even name chemicals, but it tells you what it can do. That's right. Don't forget, the bacteria don't have to do all these fancy little steps. They can just flat out release types of chemicals that kill them in the cell colony. That gets nasty. And I'm out of time. I'd like to tell you that.